in the paper 6 because it's very easy to score. Just need to follow some of the guidelines. Okay, so let me guide you on how to write the perfect answer for paper 6. Okay, let's start. Okay, I'm sorry for being rushed, but I just send you the documents. Okay, you can open the documents, which is this one. Okay, the IGCSE paper 6 notes and the paper 6 example. Okay, let me show you. Yeah, let me show about paper 6 notes. Okay, so actually the paper 6, right, is something like this. Okay, let me show you actual exam for the paper 6. Okay, so it's just like this. Okay, for example, I show you. Okay, this is on the 2000. Oh, I haven't recorded the video. I'm so sorry. Okay, I do record it. Okay, so this is on the 2025 May June. Okay, and then this is the paper 6, which with the course, okay, 6-1. Because you have about 3 variant, right? So you have 6-1, 6-2, 6-3. It depends on you will get which variant of the paper. And then, yeah, you need to answer all of the questions. Okay, answer a question and then read on the instruction first. Because I have to consider that you haven't tried paper 6 before in your school. Okay, so that's why you need to read. You need to read through the instruction very carefully. Okay, like use a black or dark blue pens, or you may use the pencil. Okay, you can use pencil to draw the diagram because it must have at least one question come out with the diagram. You need to draw them out. Write your name, central number. Okay, this is you already know, and then write your answer to each question in the space provided. And then do not use an erasable pens or correction fluid. This means what? You have the request what? Correction tape. Whenever you write the wrong answer, right, you will always want to use a correction tape to delete the wrong answer. So it's not encouraged to use this. If you write the wrong answer, cross them and then write a new sentence, okay, or write the new answer. And then remember to bring your calculator. Okay, please remember to bring your calculator. And then you should show your working and use appropriate units. So the total marks for this paper is 40. Total mark is 40. Like the paper 2. Okay, paper 2 total mark is about 80. So in this total mark is 40. But at the end, you'll be accountable weight is about 20% only. So it's just like this. Okay. Let me go through you. So you need some calculation. Okay, let me show you the actual one. Okay, actual one with the already have the answer attached on it, so it will be better for you. Okay, so you can open this one, paper 6 example. Okay, let me show you. Because I'm opening in this way, so a bit blur. I think I better open like this. Okay, paper 6 example. Okay, you can open this documents.
Yep.
Okay, so this is the page. First question. Okay, so it's like it's like this. Our question is like this. A student investigate the effect of temperature on the rate of rising bread dough, and then there will be few steps list down here, and then you need to read through it very fast. Okay, because you need to identify which is the variable. You need to identify 3 variables: the manipulated variable, responding variable, and the constant variable. This one, okay. These 3 variables, you need to identify which one is it. Because they will ask this in the questions. So let me show you. Okay. So this experiment is about what? It's about the rising of the bread dough by using the yeast, right? So it's supposed to be below to what? Aerobic or anaerobic respirations. And that's why if the yeast, right, they do the fermentation, is belong to which one? Is belong to aerobic respirations or is belong to anaerobic respirations? What do you think?
Anaerobic.
Sorry?
Anaerobic.
Yeah, anaerobic, correct. Yeah, it's anaerobic. Yeah. Okay, so that's why this is a step. First they will mix the ingredient together, which is water, sugar, yeast, and flour, and then the student use their hands to divide the dough into two pieces of similar size. You can imagine the process, okay? And then they'll place one piece of dough. You know, okay, because now you just separate it. So you have two pieces of dough. So one dough they put in the plastic cup labeled with C, another one piece they put into the transparent plastic cup labeled with W. And then they push each piece of dough to the bottom of cup. Yeah. And then the student measure the height of the dough in each cup. They took their measurements from the base of the cup to the highest point of dough like this. Yeah, okay, like this. They are actually measure the height of the bread dough for each C and W. Okay, and then later, one cup put into the cool water bath, one cup put into the warm water bath, and then the cup were left in the water bath for 10 minutes. And then after 10 minutes, the cup will be removed from the water bath, and then they will calculate, okay, they will measure the height of the dough in cup C and cup W. So you know it's anaerobic reactions. So here's the equations, okay, which is from the glucose, and it is an incomplete breakdown of glucose to become, it will release the carbon dioxide. So the carbon dioxide will cause the dough level to rise up, increase their level, and then together they will also produce the ethanol. Okay, this C2H5OH is ethanol. So then what will happen? Yeah, so this is the height, okay, of the dough in step 4 and step 6. This is the initial one. Okay, you have an initial, and then second picture is the final. Okay, the final height. Yeah, you need to measure it. So you need to bring your ruler. Don't forget your ruler and calculator. These two things. And then they ask you to prepare a table to record the result of this investigation in the space provided. Okay, measure the maximum height of dough in each cup, and then draw the line show where you took each of the measurements. So that's why you need to, okay, this is a table, right? You see how they write down the instruction. They just ask you to prepare a table, record the result. But they didn't ask you to, I mean, they didn't really ask you to give you an instruction like, okay, you need to measure the start height and the end height. Yeah, they didn't write as clear as that. So that's why you need to figure out what they really want. Okay, so that's why we look at this picture, right? Okay, you can see it's really starting of the experiment. So this is initial height, and then this is a final experiment. So you can see the W, the height is drastically increased, and the C still remains almost the same one, right? So that's why you need to measure it. Okay, by the measurement, you can get the answer. You can get the answer, and then you can assume, okay, what really happened? So this is based on the actual measurements. You just imagine you bring a ruler, right, and then you measure by the ruler. You can draw a line at here. Measure by the ruler. Okay. Yeah. So that's why this is a cup. Okay, one cool and another cup is warm, and then this is a start height. And then remember to put the unit mm. The unit start height mm, and then the end height mm. So you get the this is the data that you get from the measurements. So initial height is 36, and then the end height is 45. Slightly increase. Okay, there's increase. But for the warm card, initial height is 39, but the end height is 67. So just draw a simple table like this. It will be very, very I mean, just as simple as that. And then they ask you to calculate the change in the height of the dough in cup C and cup W between step 4 and 6. So that's why you need to minus it, okay, because you want to get whether the height is increasing or remain constant or decreasing. So the result you get is both cup also show increasing in the height of dough, but you can see that the cup, which is labeled as W, right, it will have a greater increase in the height of dough compared to the C. So that's why it's about 28 millimeter. Okay, because you need to 37 minus 39, so you get 28 millimeter. And then this cup C, right, okay, is 45 minus 36, you get 9 millimeter. Or you can write cm also can, depend on you. Okay, so the lesson is, remember, include the unit in the table. It's very important. Okay, where you can get the unit? You can get the unit from the measurement, cm or mm. Okay, because the ruler scale only has cm and mm, right? Unless in the question, right, they do give you other kind of unit, something like the micrometer or something like the meter, that kind. So that's why you need to read the question very carefully. Yeah, and then you need to state a conclusion for this result. Okay. Oh, at the higher temperature, which is the cup labeled with W, the dough rise more due to because okay, higher temperature, dough rise more. Okay, why the dough rise more? Because it has a higher production of carbon dioxide, because more carbon dioxide is being produced in the cup labeled W. So that's why more carbon dioxide, it will cause the dough rise more, increase in their height. So this is the conclusions. And then see, we need to identify which one is independent, which one is dependent variables. So the independent variables, you can see they are changing for what? So that's why you need to find the answer from those statements. Especially in the first sentence, you can see the first sentence, right? They are investigating the effect of temperature on the rate of rising of bread dough. So effect of temperature, they are changing the temperature. So that's why the manipulated variables is belong to the change in the temperature. Change in the temperature, okay? And then another one, responding variable. So you can write, okay, temperature. You can see the change in temperature, something like that. And then you need to identify the dependent variables. The dependent variable is, they refer to the variables that will measure in this investigation. But just the dependent variable, right, is a bit tricky, because when you write the dependent variables, usually in the question, right, okay, usually in the question, they will give you the manipulated and the responding variable in the sentence, but in the manipulated variables, they will just directly give you which is the answer, temperature. But another one is responding variable, they will not directly give you the answer. Okay, so they'll just give you like this, the rate of rising the bread dough. Okay, so this is what, and then you already know that this is referred to the responding variable. But in the answer, right, in the answer, what do you write? Okay, you cannot write the rate of increasing in the height of the dough. You cannot copy from the questions. So that's why you need to write the dependent variable based on what you have did. So just now you are actually calculate the height of the dough, right, which is this one. You actually calculate the increasing, okay, how much increasing in the height of the dough. So that's why this is the answer that you should write. Height of the dough, because you are calculating them. Okay, so that's why this is the question. They state what? They just state that the rate of rising bread dough, but they didn't ask you, I mean, they didn't give you information that usually you need to calculate the height of bread dough by using the ruler, something like that. So that's why usually the manipulated variable, you just copy from the questions, but the responding variable, right, you need to think, you need to think back what you actually do in this experiment. Okay, so that's all. You can write height of dough, or you can say that distance from bottom of cup to the top level of dough also can. Okay, so, and then the second question, okay, identify one possible source of error in step 2 and describe how the method could be improved to avoid this error. Okay, this is also common question, okay? They must ask you this kind of question. So the answer is already fixed. How you able to reduce the source of error? And then how you able to improve the accuracy of the result? Because, right, the error, right, they refer to any kind of variables. They can, I mean, they will affect the result accuracy. For example, if you use your hand to separate the dough into 2 pieces, you just use your hand to separate it. You didn't use any weight scale to measure it out whether it's really they have the same mass. You didn't do this you didn't do this procedure, right? So that's why the error might be due to the dough was not accurately divided into 2 parts. So might be one dough is lighter, one dough is heavier in their mass. So that's why this is your improvement that you need to do. You actually need to measure the mass of dough by using the electronic balance to ensure both dough, they are recorded with the same mass. Yeah, so this is improvements. Oh, and then this one, explain why a more valid conclusion can be made by comparing the change in height rather than final height of the dove. Okay, why you need to compare the change in height? It's because that even though it's starting from the initial height, right, which is this one, which is the step 4, even though it's from initial height, but the C and the dove level in the C and W, right, they might show difference. So you can see, right, dove W, right, is slightly higher than the dove in the C. Maybe they separate the dove, right, because they didn't measure it, they just separate manually by the hand. So that's why the dove is being separated in the uneven side. So it will cause the cup label with W, they have higher mass of dove compared to the mass, compared to the dove in the cup C with a lower mass. So it's better if you can estimate the change in the dove level. So you can measure that which dove actually increase the most compared to other dove. So you should write in this way. Where is it? So this is because that the height of dove at the beginning, was not identical in both cups. Because starting point, they already level is not different. And then you still remember in the chapter 4 biological molecule, we learned about the food test. And then this food test, right, is very common questions to come up in paper 6. You need to remember the color of the food test. So for example, that they ask you this one, okay, the color changed when the starch was present in the sample of bread dough. So give a positive test result. So the test is built to what? Iodine solutions. So a positive result is blue-black color. Okay. And then for second one, okay, the dough mixture in step 1 contains 50 grams of flour, 3 grams of sugars, and 2 grams of yeast. And then they will produce a carbon dioxide when the yeast do the anaerobic respirations. So guess, the carbon dioxide would cause the dough to rise. And they also state the number indicator, which can prove that the gas produced is belong to the carbon dioxide. So the indicator, okay, they use is actually this hydrogen carbonate indicator. And then this is very famous indicator to test whether there's a presence of the carbon dioxide. And then, okay, at this one, carbon dioxide is the acidic gas or is neutral gas or is alkaline gas? Which one? Hello? Carbon dioxide is acidic gas or alkaline gas or neutral
Acidic
gas?
gas.
Yeah,
Acidic.
it's acidic gas, correct. It's acidic gas. So you still remember, right? In the respiration, we learn about what? If you do the vigorous exercise, you inhale. It's not you inhale. Okay, you do the vigorous exercise. So you actually increase the rate of breathing. Because you need to supply more oxygen to your muscle, to your working muscle, to do the aerobic respiration. So your muscle they can request what your muscle cell, they can do the cellular aerobic respirations, so they can release more energy to supply them. So that's why by this way, right, they are doing the cellular aerobic respiration, so they will produce a lot of carbon dioxide as a byproduct. So that's why this carbon dioxide will be released to the bloodstream. So that's why if you do a vigorous exercise, okay, you will cause the effect, which is you increase the level of carbon dioxide in your bloodstream. So that's why your bloodstream become more acidic. Okay, because there's an excessive amount of carbon dioxide being produced and they will be accumulated in your bloodstream. So that's why your blood pH, right, actually reducing, right? Because the carbon dioxide is acidic gas. So if you have too much carbon dioxide in your bloodstream, so it will cause the blood pH to reduce from 7.45 or 7.35 will reduce to become 7.2, something like that. Okay. So pH 7 to blood pH with 7.2 is not good for your health, and then it will cause something like detrimental effect to your health. Okay, you will feel very tired, something like that. So that's why your body will working hard to remove the carbon dioxide away by what? By increasing the rate of the ventilation or exhalation. Okay, so they will exhalate to remove away the carbon dioxide from the bloodstream through the exhalations. Okay, so that's why the hydrogen carbon indicator, okay, they have about a range of color from pink become yellow or orange. So if the hydrogen carbonate indicator show that it's in pink, right, that means the solution itself is acidic or alkaline. What's the answer? If the hydrogen carbonate solutions, sorry?
Acidic.
Or it's in pink. Pink referred is in alkaline. Let me show you this one. Hydrogen's carbonate indicator. Okay. So pink means it's alkaline. Which is this one. Pink or purple? Oh, what happened? Okay. Let me show this one. You can see, right, Edder Suyang? Remember this image? Where should I put? I think I put in your group. Okay, I put in the group. So you can see the image show about what? See, this one. I think I put here. Hydrogen carbonate indicator. Remember, if the solution itself it is alkaline, the indicator will change to become purple or red color because it's alkaline. See? But if the solution become more acidic, increase in the acidity due to what? Due to we are increasing in the carbon dioxide concentration in the solutions. Yeah, so it's like the pH indicator they will change to become more in yellow or orange in color. So remember, if the color is showing red or pink or purple, that means it's alkaline pH. If the color show that it's yellow or orange color, they show it is more to the acidic pH, which is referred to they have more carbon dioxide in the area. So that's why if the gas is carbon dioxide, if there is a presence of carbon dioxide in that area, so it will change the indicator color, become yellow or orange color. You can say yellow color or orange color, up to you. But you cannot say red color, purple color. No, that's color belong to the alkaline solutions. Okay, and then this one, 6 months. Okay, this is also they have a fixed procedure. You need to plan an investigation to determine the effect of mass of sugar on the volume of carbon dioxide produced by the yeast suspensions. So which one? Okay, from this statement, right, you need to identify which one is your independent variable and then which one is your dependent variables. So effect of mass of sugars. So they are referring to what? They're actually changing the mass of sugar, right? So the mass of sugar, they can be lesser or they can be more sugar amount. So how this mass of sugar they will affect, they will cause the effects on the volume of the carbon dioxide produced. So that's why the mass of sugar is belong to what? Is belong to the independent variables because you can change it, you can change this variable, and then you can see how this variable they can affect on other variable, which is the volume of carbon dioxide produced by the yeast suspensions. So that's why independent variables you can refer to different mass of sugar or amount of sugar being used. And that's what I mean that if you write the variable, right, you also cannot copy the title. You cannot write that independent variable is mass of sugar. That's all. Okay, you should add on something like you are actually using different mass of sugar. You are actually using different amount of sugar. Write it in a more, clear. It's not detailed. Yeah, you need to be more detailed in your answer. And then the method is what? Okay, the method, you need to design your own method. Design your own method, okay? Putting the dough into the same size cup and container. Then after that, measure the height of dough. Or you can because you are actually measuring the height of dough because you want to measure the responding variable. Or you can use another kind of method, which is you can measure the production of carbon dioxide gas. Okay, but another thing is very important is when you write the method, you use what kind of method to measure your responding variable. For example, I use to measure my height of dough, right? By using what? By measuring the measuring cylinder or ruler. So I can measure the change in height of dough. If I want to measure how many carbon dioxide gas is being released out, I can use a gas string. I use a gas string to collect it, to collect my carbon dioxide gas. So if more carbon dioxide gas are being released, that means the rate of reaction is higher. Okay, so that means the rate of the anaerobic respiration is higher. Might be due to more glucose. I mean more sugar being used to carry the experiment, so you can release more carbon dioxide. Because it's more sugar, right? More sugar, they can allow the yeast to have a higher rate of the higher rate of the anaerobic respirations. Now, so the method is about how you able to measure the responding variable, and also how you're able to prepare the ingredients. Prepare ingredient, and then how you use any kind of method to measure your responding variables. And then, okay, the dependent variables is measure volume of carbon dioxide produced in a set time, okay, or in a specific time periods. Because you need to measure the products in a given time period. So something like it's like 30 minutes or 1 hour. You cannot be longer or shorter. And then you need to standardize for all experiments. You are actually using the same time frame. So that's why you can measure the volume of carbon dioxide produced in the set time or in a given time also can. And then the constant variable refer to you should use like same type or same source of yeast. Okay, and then you also can say you can use same type of sugar, which is brown sugar or white sugar, fix your sugar. Measure of time to produce a specific result. Okay, this is also known as constant variables because you use all the same time frame, okay, which is the experiment must be conducted, must be measured in about after 40 minutes or after 1 hour. So in a 10 minutes, you need to measure the result, okay, and then stop it. And also pH, because pH will affect the conditions, okay, in the anaerobic respiration. Because for the yeast to conduct the anaerobic respirations in that given situation, right? Okay, the pH cannot be too high or too low. If there's any change in pH, they will affect the yeast activity also. So the yeast might become inactive, so they can produce lesser carbon dioxide. So that's why we want to prevent this happen. We need to fix all the pH. All the pH, the test sample, the pH become same. So that's why pH is another indicator. It's another constant variables. Okay, because remember, you know, this one, okay, which is this one, the aerobic respirations. Sorry, it's anaerobic. Okay, this one. The anaerobic respiration in the yeast, right, actually is enzyme catalyzed reactions. So any kind of reactions which is being catalyzed by enzyme, they will be very sensitive in change in temperature or the pH. Okay. Oh, yeah. So this is the way on how you write it, and then the procedure is already being fixed. So you need to include your independent variables, dependent variable, and constant variables. List down them, and then list down them, and then for the independent variables and the dependent variable, right, you can write one enough already. Okay, for example, this one, independent variables, you just write different mass of sugar, stop. Or you can write amount of sugar used. Just write one sentence is enough already. Same to the dependent variables. They just measure volume of carbon dioxide produced in a set type. Just one sentence, 1 point. But the constant variable, right, you can write more than 1 point. You can write about 2 point or 3 point maximums. Okay, so that's why I write source of yeast, pH. You can write more than one. And then the method, okay, method is you should write by using what? Any kind of apparatus to help you to get the result. For example, you are using the gas string or you are using the measuring cylinder. So this 4 point already you have, right? The 3 variable, the method. And then another tool is what? Another tool is already fixed one. you need to improve your result accuracy, right? So at this one, how you're able to improve your result accuracy for the experiment? Very simple. Just repeat it. Repeat the experiment, and then you can get more data. You can build up a database. So from this, you can have more accurate test results. So just repeat. So repeat the experiment for another 2 times. Okay, that's all. So this is the best way you're able to increase the test accuracy. And then how you're able to protect yourself during experiments? Because they ask you to plan and procedure, okay, or investigation. So how you're able to plan it, and then how you're able to protect yourself while you are performing the experiment. Because experiment, they might be very, very dangerous. I think, and that's why you also did the experiment before, right, in your school. Okay, so you are doing what? You're doing the Bunsen burner, the kind of chemical substance, so it might be very, very, very dangerous. So how you can protect yourself? You can wear the goggles, the eye goggles, or you can wear any eye protection during performing experiment. Yeah. So there's a 6 mark being given for those answers. Okay, the 3 variable method, how you improve the accuracy, repeat the experiments, and then how to protect yourself. And then another thing is this one. Okay, usually they come out about 2 questions for the paper 6. Okay, so this is the and then they will come out a lot of picture. Usually it's about picture, and then they ask you the information from the picture. So this image show there's several cells from the Elodea aquatic plants. Elodea is referred to the plants just grow inside the water. And then draw a large diagram of cell label A. Okay, so this is also considered as a gift mark to you. Okay, a gift to you, because you just need to know how to draw it, and then you can score it. And then you hear how many marks? 3 marks. I think it's 3 marks. Oh, 4 marks. Okay, 4 marks. So how you can draw it is, they ask you draw a large diagram of a cell label A in the figure 2.1. Just draw a very large diagram. How large is it? Okay, remember your paper. Okay, at this one, your paper is just like this, right? Like this layout. Okay, you should draw your diagram at least half of the given blank. So your given space is like this, right? So you should draw the space occupy more than half of the space. So you should draw like this. Okay, which is occupy almost half of the space. You cannot draw too small like this. Cannot. Okay, I hope you get what I mean. Ensure your drawing occupy half of the space. Okay, draw as bigger as you could. And then another thing is, whenever you draw a diagram, the thing that you need to note down is, okay, they ask you to draw this cell label as A. So you just follow them, draw the outline, a clear outline like this, okay? A clear outline. No shading. Because you are answering the biological question, okay, you are not an art student. You are not doing your artworks. If you do artwork, you use a shading but you are actually answering this paper 6 biological question. So that's why you just need to draw a clear outline. Use a pencil. And also remember to use a pencil, okay? Don't use the pen, because if you draw wrong, it's very, very troublesome. Use pencil to draw it. Even though you draw wrong, or you feel not satisfied, just erase it. Redraw again. So usually they will just use pencil for these sections. Draw it. Okay, draw a large diagram. So just ensure clear outline, no shading. And sometimes if the question asks you to do what? If the question asks you to label, and then you need label. Okay, for example, let me show you this one. Okay, so this is another template. Okay. Okay, I show the drawing part. Oh, see the drawing part. Okay, use more than third of the fourth of the space given, which is you need to use more than half of the space. Cannot draw too small. And then make sure you draw with the pencil, so it's easier to erase. Okay, I think there's one time a student they draw with pen, and then what happened? They draw with pen, and then they feel like not satisfied with the drawing, and then they use the what correction tape to correct it all, and then it will be very, very messy. Yeah. So that's why use pencil. And then you need to have has a definite outline. Outline just like this, okay? A clear outline. Just a clear outline. 一个简单的线条啊。clear outline refer to what? 简单的线条，标打阴影。And then no arrowheads when labeling. Okay, when you do this kind of labeling, right? You shouldn't put the arrowhead. 不要放箭头啊，一个直线而已。Okay, unless if the questions ask you to draw arrowhead, or only you draw the arrowhead. But if they don't ask you to draw arrowhead, they just ask the label. So you just draw a simple line pointing to the image like this. See? Okay. This show about what? Ovum and the one sperm cell. And then they ask you to make a large drawing of ovum and the sperm cell. Label the ovum. This is the thing that you write. Okay, so this is a model answer. Okay, just draw a simple line and then with the sperm attached on it. 4 marks. Okay, so how they can award you with the marks? So first mark. If you draw the single line, okay, single line, give you 1 mark. If you didn't shade your drawing, okay, another mark. And then another is you draw half of the space given. Okay, there will be the mark. And then you draw with correct proportions. Correct proportions, understand? 正常的比例. For example, you know that ovum is larger than sperm a lot, right? The size of ovum is definitely larger than sperm. So you should draw out also at this way, which is the ovum is larger than sperm. You cannot draw the ovum and sperm is same in their size. It will be very, very weird. Correct. Correct proportion is very important. And then how you label it? Label the ovum. So you can see this is how they label it. Just use a ruler to attach the ovum. That's all. Cannot draw the arrowhead. I just worry if you draw the arrowhead, even though your answer is correct, but the examiner, they might deduct your mark, and then you lose 1 mark. It's very unfair to you, right? Okay, so just draw it like this, okay? And then they might give you different kind of image, and then something weird image. So that's why this is just draw in simple line. And then you should do this very fast, because it's a simple questions, okay? You just need to follow the instruction given. Yeah, like this. Just follow the question, okay? And that's one. If they don't ask you to label, please don't label. Just draw it. If they ask you to label, only you label. Everything follow the questions. Don't add additional thing on your answer. Okay, and then this one, okay. Measure the length. So this is another questions. So in this question, right, they already give you with the multiplications of like this is times 1,200. And then measure the length of line PQ. So this is usually you use a ruler. Okay, so use a ruler, you measure, right, you get 93 millimeter. And then you need to calculate the actual length of the cell using the formula on your measurements. So multiplications, okay, the formula, okay, which is the 1,200 equal to 93 millimeters, which is because this 93 millimeter is your image size, so you need to know your actual length. So over this one. Sorry, my handwriting is not good. But in the actual exam, right, you should write a neat answer, okay? So that's why you get 0.08 millimeter. So write your procedure. Okay, this one is what? Actual length of cell, okay? Actual length of cell. Yeah, so 93 millimeter over 1,200. So we get 0.08 millimeter. So your answer is 0.08 millimeter. Include the unit, okay? Don't forget your unit. So you get 3 marks, like this, 3 marks. So this should be very, very easy for you. And then for this one, another question is, a student investigate the effect of light intensity on the rate of photosynthesis. So they give you another questions, which is in the Elodea species. And then they change the light intensity by placing a light source at different distance from the plants. See, different light intensity, different distance from the plant. And then you calculate the number of bubble of oxygen produced. So this should be the responding variable, right? And then the one is a manipulative variable is referring to the light intensity and also the distance, okay? Distance between the light source and the plant. Yeah. So this is your result, okay? Given in table. And then what you need to do is calculate the rate of bubble productions when the light source was 120 cm from the plants. Yeah, so this is a distance, okay, and then this rate of bubble productions. Yeah, so you get about 103 bubble per minute. Okay, how you get a 103 bubble per minute? Is by the calculation, okay. Where I do show you at here. Oh no, I didn't show you at here. Okay, which is you refer to this one. Okay, you have about 80 cm of the light source from the plant. Okay, so from the 80 cm of the light source from the plant, right, you increasing to 100s and then to 120s, and then you can see that when you increasing the distance of the light from the plants, and then what will happen means that the plants, right, they become more further away from the light source. Plants further from the light source. So it means what? It means that the photosynthesis rate is actually reducing, right? Because the plants, they absorb lesser amount of light because the light is just further away from them. So that's why the number of the bubble produced in 3 minutes is about 309. Yeah. So this is just very simple, okay? 309, you just divide by 3, you get 103. Okay, and then usually they will ask you to plot a line graph. So that's why it's very important. You need to prepare your rulers. Plot a line graph on the grid, show the relationship between the distance of light source for the plant and rate of bubble production. So identify which one is your x-axis, which one is your y-axis. So at this one, what do you think? Which one is the x-axis? Which one is the y-axis? Distance of the light source from the plant is the x-axis or y-axis? I think I should. Yes, okay. It's in the x-axis, which is this one. Yeah. So that's why you need to draw a line, okay? And then you need to draw your scale here. And then you need to design your scale like how to say a bar, okay? A bar is equivalent to how many units? So for example, you can see the distance, right? See, from 80, 100, 120, and then you get until 200s. So you can decide that maybe every one scale bar, you can put about 20 units. Okay, like this, okay? So the 20, 40, 60, 80, and then at the end you get 200 units. So it's still enough for you. So usually in the exam, right, they will just design the area of the the area which is just enough for you to draw a line graph. So that's why it's very important that you need to design the correct unit. Because if you put about like 50, 100, 150, it will be very weird, okay? Because the graph might be very, very small. And then when you draw this graph, when you draw the graph at this one, the graph must be occupy half of the given area also. You cannot draw a graph which is too small. Even though your skill, your concept is correct, but the graph cannot be too small. They should be like this, just very big, clear. They can show each of the data in a very clear way. So that's why X, okay, which is referred to distance of light source from the plant, dash with the units, okay? Remember to write your unit on the x-axis, okay? Just everything you need to include the units. And then this one, rate of bubble production per minute, which is the y-axis. Also, you put the skew 10, 20, 30s, 40s, yeah, the skew. And then what you need to do is you need to plot a line graph. What's meant by line graph? Line graph is referred to you use a line to connect the dot together. Yeah. So that's why this is the result. Okay, this is the result. Yeah, starting from where? Okay, they are actually starting from the okay 20, 40, 60, right? So start up here. Okay, 80. They just start from 80, 80 until 200. So by using the figure here, they are using the figure here. So that's why this is the line graph that you get. Okay. And then yeah, so that's why drawing a correct line graph is very important, because the next question is they ask you to use a line graph to estimate how many is the rate of bubble production per minute if there is a distance from light source of 170 cm. Okay, so 160 cm, right, is referred to here already. Okay, you got 20, 40, right? It's at least around here, okay, around here. Yeah, so that's why it's around here, okay, 170, yeah. So that's why your line graph, right, you should touch the y-axis. So that's why the answer they get is 103 bubble per minute. Okay, 103 bubble per minute. And then this one, this one the trend show in the graph. So as a plant move further away from the light, the number of bubbles remain constant and decrease. The trend show in the graph. This is just something like in the paper 4, there's a questions. They ask about what? Describe and explains, right? And then describe, you need to describe the trend of graph, whether this graph is showing increasing or decreasing or plateau. So just something like this, describe the trends. So as a plant move further away from the lights, the number of bubbles remain constant. Initially it's remain constant, but later they will decrease, which is here. Okay, see, initially they remain constant like 3, 1, 2, and then decrease to 3, 0, 9, and then to 2, 7, 2, 6, 4, and then they decrease more again. So remain constant, later they will decrease. These are the trends. And then they are changed in the rate of 100 to 130 cm after 2 readings, which is here. Okay, so after 2 readings, right, you can see the number of bubble is actually showing there's a change. How many marks? They will give you 2 marks. So that's why you need to write at least 2 points. Okay, and then for this one, the student repeat the experiments described at a higher temperature. The student left the plants for about 10 minutes at the new temperature before starting to measure the rate of bubble productions. And then suggest why the students wait for 10 minutes before taking measurements. Okay, why they need to wait for 10 minutes first? Only they will do the measurement. So because the reason is, if they didn't wait for 10 minutes, right, they just took the measurements in first minutes or second minutes. So what the condition that can happen is they might be, okay, this refer to what? Okay, this refer to if you repeat the same experiments at a higher temperatures, okay, and then you left the 10 minutes at the new temperature, which is higher one, before starting to measure the rate of bubble productions. Because if you change the temperature, right, and then this photosynthesis, the photosynthesis process is being affected by enzyme also. If you change the temperature, the photosynthesis rate, they might be increasing also. Okay, we assume that photosynthesis rate will be increasing. Yeah, so that's why you are actually changing the conditions. You are actually changing the condition provided to the plants, to this Elodea plant species. So that's why you need to wait first before you take the measurements. So this is for you to equilibrating to the new condition. You need to give time for the plant to adapt first, adapt to the new condition provided to them first. Only they will like stable, and then they will start to do their reactions. Because if you change the temperature, right, from low become higher temperature, the plant also will have a shock like that. Okay, so that's why they need some time to adapt to the conditions. So the answer is idea of plant equilibrating to the new conditions. Okay, and then this one, okay, another calculation, but just a very simple calculation only. Yeah, so this table 2.1, they show that the light source was 140 cm from the plants, and then the rate of bubble productions was 88 bubble per minute at the origin temperature. So at the higher temperature, the rate of bubble productions when the light source was 140 cm from the plants was 122 bubble per minute. So they ask you to calculate percentage change in the number of bubble when you are actually increasing temperature. So in the origin temperature, which is, it didn't stay also, but you know that in the origin temperature, right, is 88 bubble per minute. 1 minute, it can release about 88 bubbles. But if you increase the temperature, become higher. So now they'll become what? 122 bubble per minute. So we need to calculate, so that's why you need to calculate the change in number of bubble. So how do you calculate? Just remember the formula, final data minus initial data. So that's why the final data is 122. 122, you need to minus the initial one, which is 88. And then you need to over the initial data, which is 88, times 100%. So at this one, remember to write the 100% in your answer, because your answer is in the percentage. So you can see, right, it's in the percentage. So that's why you need to write the answer in the percentage form. So the answer is about 39, okay? 39 percentage. So actually it's showing about what? 39% increase in the number of bubbles when you increase temperature. So that means the kinetic energy for the enzyme, for the substrate become increased. So that's why the substrate and enzyme, right, they can collide more frequently. So you can increase the rate of the enzymatic reactions. Okay, finish, that's all. So that's why in the paper 6, right, it's very, very easy to score. And then usually it's a part that will help you to mark up the score, whether to A or A+. So generally just like this, okay? So let me show you one by one, okay? One by one on how to answer the question. Actually, there's a format. Okay? Let me guide you to this document first. Okay, so this is the paper set revisions. Okay, this document is, I also send to WeChat, which is here. This one. IGCSE paper 6 notes, okay? So you can open this document, the Word document, and you can have a look. So that's why when you want to make a units, and then because sometimes, right, the title they will give you, I mean in the question, right, they are stated in the minutes, okay, but in the table they ask you to write the answer in second. So that's why you need to convert the unit become second. Okay, so they will give you some of these measurement units. Measurements question like this. Record the volume for DC battery remaining in the strings. Okay, so you have a hot test tube and cold test tube. Just need to measure their volume. Okay, so you just use a ruler to measure it. Okay, so this is the answer that you get. Okay, so one is about 3.5 cm3. Another one is for 1.0 cm3. Yeah, and then prepare a table to record the volume of DC PIB that have been used in each test tube in each space provided. And then use your answer in the equation to calculate the volume of DC PIB that has been used to each test tube. So you know that the function of DC PIB is to detect the concentration of this vitamin C in the solutions. Yeah, they want to detect the vitamin C in the solution. So it's like this is the data that you get. Okay. Which is you just follow the formula. Okay. Sometimes they will just give you a formula like this, which is the volume of DC PIB used, which is a 10. 10 is being given formula, right? So it's like you 10 minus the volume of DC PIB remaining in the string. So if you have a look on the hot, on the hot string, right? So maybe it's about this unit, right? Is about 6.5. Yeah, it's about 6.5. Given is in the cm3 because it's volume. Yeah, so volume you should write in the cm3. So you can write 6.5 cm3. So tens minus 6.5 cm cube. See, 10 minus 6.5 cm cube. So it's about 3.5 cm cube. Same to the cold one. So initially your length is not your length. Initially your measurement, okay, is about 9 cm cube. So 10 minus 9 cm cube, you get 1. Okay, 1 cm cube. And then so on. Whenever you draw about the figure, right? Oh, you can see they didn't write as 1. They write as 1.0. Why they write as 1.0? Even though this question they didn't ask you to something like draw your answer in 2 centimeter figure, but if they ask you to draw your answer in 2 centimeter figure, right, you should follow the instructions. Okay, but in here they didn't ask you about this one. So that's why you should write the answer. Okay, 1.0, because your upside answer, right, is 3.5. So it's about 1 significant figure. So that's why you cannot put 1 on it. You should put 1.0 to standardize. Okay, standardize the measurements. Okay, and then for the graph, how you're able to draw a pretty graph? So you have the x-axis and the y-axis. Okay, so x-axis is always belong to the independent variable, which is you're actually changing, modify on the variables, because independent variables. And then dependent variable is referring to you use what kind of method to measure the variables. And then skew. When you draw the graph, right, usually the skew, they always start from 0. Starting from 0, okay, which is here, okay, like this. Where is it? See? For example, this is a graph, right? They just start from 0. So that's why you need to adjust your unit. Okay? Adjust the unit here. Same to your x-axis and also the y-axis. You need to adjust the unit. So you need to determine that like 1 skew bar equal to how many units? Is 20 units or 50 units? Occupy half of the space, okay, for the graph. So you can see, right, this graph is very clear. It's very clear to have a look on the trends. So you can see, right, the trend is showing that increasing initially until they reach a peak, and then it's decreasing. So it shows you the trend very clear. So you need to adjust the skew so the graph fill more than half of the space even. And then do not use odd spacing. What's mean by this? When you draw the skew, right, you better to use an even number. 2, 4, 6, 8, 10. It's very rare to use the odd spacing like 1, 3, 5, 7. I think no one they will use it. So it's like better to use even number. Start with 2, okay, 2, 4, 6, 8, like that. Because you are drawing the line graph, right? So it's very important that you should join the point together by the pencil. And then you need to describe the graph. So talking about relationship between the independent and dependent variables, use a figure in the answer. For example, they show you increasing trends, and then they reach a peak of the and then you can input the data. So just like how you're able to answer the paper 4, it's almost the same thing. So you can see, this is the table, okay? So usually they will just give you a table like this, with the temperature and the rate of reactions in each of the products per unit time. They give you the parameter and also the units together. So that's why whenever you draw the graph, right, you need to include the name of the x-axis, okay, which is in here. There should be temperature. So you can write temperature and then with the unit here, okay? See, remember to include the unit, because if you didn't include a unit, even though your parameter is correct, but the examiner, they still can deduct your mark because you didn't write unit. Unit is very important. So you should write unit for your x-axis and also y-axis. Just include unit at beside, okay, beside of the parameter. Okay, so line graph is just one of it. And then line graph is very common to come in exam. Another one which is less common one will be this one, histogram. Have you heard of histogram before? Histogram, okay, what's by histogram? Why it didn't show in the picture? Here, Okay, so this is a histogram. And then histogram, right, will be different from the line graph because histograms they are referring to, you actually need to compare. You need to compare about two groups. Usually histogram will come in this way. You need to compare the two groups. For example, you have about men and women, right, with the same age group. Okay, so 65 to 74. And then you can say, whenever the age group is increasing, it become more older. So there will be also increase in the percentage for the anemia. So you also can do a conclusion something like the risk of anemia is increased with the age. So that's why you need to plot the histogram. Yeah. That's why histogram is like this. Okay, you need to compare about two groups. One is men, one is women, with the same parameter, which is the age group, right? So that's why from 65 to 74 is like this. Okay, here. Okay, so later 75 to 85, right, will be here. And then 85 to 90 will be at here. So you have about 3 age group. You have about 3 age group. So right, and then you need to identify which one is man, which one is woman. So that's why man is below. Okay, so here is a man below. And then here's a woman above. So that's why whenever you draw the line, right, for the men's, it shows about 7.0, but the woman shows about 8.5. So that's why 7.0 plus 8.5 is about 15.5, right? So that's why your skill for the woman located at 15.5. Same to the second group, okay, which is 75 to 84. And then the main group is around here, 16.5 here, which is above 15. And then you add on with the women's, okay, so 16.5 plus 11, you become 27.5. So that's why you get to here. And then the line graph, how they draw is different from this histogram. So you can see that histograms, you need to shade them. Why you need to shade them? Because and then you can just like this, okay? You see, right? They just label it. So you can just label for your own way. For example, you can write that. I just draw here, okay? The man, right, is being shaded like this. Okay, for the woman, it's being shaded like this, which is horizontal in the line. So that's why the man, okay, is like this later. Okay, how about the woman? It's like this later. So this is histogram. Only histogram they can do this, okay? They can slightly do the stripe, we call it stripe. Because we want to differentiate which group is belong to which one. And then for this one, yeah. Okay. Okay. Another thing is for the histogram, you are not required to start from 0 already. You know why? Not like the line graph. Line graph, right? You need to always start from 0. But in the histogram, you are not required to start the 0. It's because just a simple comparison about the men and the women's, how is the percentage of anemia. That's all. And then what will be the anomalous result? Anomalous result is like whenever you conduct experiment, right, you will get some of the result which is very, very far away from the average result. So this is called anomalous result. Okay, so it's referred to the result from other table or point, doesn't follow the trend of graph. And then make the comparison. Okay, I think you should able to know how to make it. And then drawing, yeah, we will do. Okay, so you can see this one. How to plan an experiment. So find the 3 variables. Okay, so usually they ask you to find the manipulated variables. They need to be changed and mentioned. How are you going to change it? Okay, and then you need to include apparatus. For example, you change your temperature by using the water bath heaters to about 10, 20, 30, 40, 50. You can write your preferred amount because this experiment is being designed by you. So it's like you can modify, okay, based on your preference. You can write about the temperature 5, 10, 15, 20 also can, up to you. Then how you are going to measure the experiment result? If you are checking the presence of starch, right, you want to measure the time taken for the food sample to turn to the blue black with adding the iodine solutions. Okay, so it means that just like you are doing the food test for starch, so you just drop the iodines on the food contain the starch, and then you check. They require how long time, okay, how much the time to change the iodine color to become yellow brown, okay, not blue black at all. And then list all the variable, okay, which is the one, the constant variable, responding, governmental variables. And then you can list down some methods, okay, about how long your experiment will last. And then this one, this is very important. And also this last two sentences, right, is super important because you just sometimes even you don't know how to answer the questions, but when you write this procedure part, which is 6 months, you can repeat this word. Repeat experiment for about 2 times to get more reliable result. This is a word you can write. For many kind of experiments, right, this can apply. I mean, this answer they can apply in a lot of experiments. Because if you get more data, you have more accurate result. You get more data, you're actually reducing the anomalies result. And then safety precautions. For example, like wear the glove. You can say wear glove, wear eye goggles. Up to you. So this is on how you plan the experiment. Okay, let me see the example answer. And this one, see. Plan an investigation to determine the effects of temperatures on the breakdown of starch by the enzyme. So effect of temperature. This is the manipulated variables. Okay, breakdown of starch by enzyme. Okay, this is responding variable. But how are you able to write this responding variable? Okay, you know it's breakdown of starch by the enzyme. So how are you able to convert it become the responding variable? You cannot just copy this breakdown of starch by enzyme, right? So it refers to how you're able to measure the rate of the enzymatic reactions. Do you have any idea at this one? Okay, it's okay. Maybe I asked you two already. Yeah. So let me show you. Okay, just imagine. Okay, now you're actually conducting experiments. Okay, this experiment is being conducted at different temperature. So in the manipulated variables, okay, the manipulated variable, you can write different temperature like this. And then how you can measure the rate of enzyme being affected by temperature. So you just measure about how many starch already been broken down by the enzyme, which is maltose. So how you able to measure it? So you measure it by dripping the iodine solutions. So if the iodine solutions change from blue-black become so just imagine this way, right? Okay. You know, sometimes they will prepare white towel like this, white towel. This one. If they want to do the iodine test for starch. Sorry, it's not this one, okay? This one is starch solution. This one. See? This is how they do it. Okay, this is very, very nice. Okay, I want to show this one. Okay, yeah. So how they do this is they're actually using this rock towel. Okay, I think in future, if you study biology, you will do this in the school lab. So it's a kind of popular experiment procedure, okay, in the biology. So you can see initially when you just drop the iodines into the solutions, right? The iodine just changed to blue blacks. Okay, because there's no digestions. Okay, but what happened if so this is different concern also. Okay, so these are the 1%, 2%. So you can see, right? If you want to compare 1% to 2%, very obviously is the percent number 2. Okay, even though they use lesser time, 8 minutes compared to the here with a 1%, 12 minutes. So that's why you can see the color. Okay, initially it's blue color. At the why initially is blue in color? Because inside they have the starch, right? Yeah, initially the starch is present, but later when the time goes by, the starch will become what? The starch will become the maltose. It's because being broken down by the enzyme maltase. So that's why when you continue to drop when you continue to drop the iodine, right? So later it will not change in color already, just remain the color. So you can see if you compare to this one and this one, which is here, if you compare to here and here, right? What will happen is, even though the first one 12 minutes, the second one's 8 minutes. 12 minutes is longer than 8 minutes for about 4 minutes longer. But what happens if this one still have a faster rate of reactions here? Okay. Sorry, I cannot find my cursor. Here. Okay. So that's why this one, they're supposed to have a higher rate of reactions. Higher rate of enzymatic reaction compared to the 1%. So do you know why? Why the 2% they have a higher rate of reactions? What's the answer? Why these 2%, right? They are being recorded with a higher rate of reaction. It's because the starch is not starch. The iodines, they remain in the yellow brown color, starting from where? Starting from here. Okay, here. So they refer to digestions of the starch take place, because what happens if there's a starch digestions? The starch will be break down, become what kind of substrates? And that's also remember, during starch digestions, right? Starch will be break down by what enzyme? Become what kind of substance? What kind of products? Waiting for your answer. Yes, your answer is
Amylase. Amylase.
amylase. Okay, amylase will digest the starch, but starch will become what kind of substance? Yes, the amylase, yeah, the enzyme.
Glucose.
Maltose.
Okay.
Maltose, no glucose. Maltose. Okay, be careful about this one. Starch can be break down, become the maltose. First, only from the maltose, right, they'll become the glucose. So you cannot just write, when you write the starch become glucose, right, it's incorrect. You should write the starch become maltose first. So in this situation, also the same thing. Why in this experiment, right, okay, the 8 minutes and 12 minutes, this is referring to what? This is referring to they actually use about 8 minutes, just 8 minutes. The wort tile, okay, the wort tiles, the color iodines, okay, the color iodines, right, they will not change in color anymore, starting from here. See? Yeah. So they just require about 8 minutes. So which is referring to the iodine solution, right? They just remain in the original color, doesn't change in color. And then this 1%, right? 1% they require what, 12 minutes, which is they require longer time, much more longer time. So 12 minutes only, which is after 12 minutes only, the iodine solutions, they cannot only, they will not, they will just remain in the color, which is here. So they require about 12 minutes. Because when they drop, right, they just drop one by one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, around that. Just drip in one by one. So they just drip until the iodines, right, they will just remain in their original color, not change, become dark blue anymore. Okay. Another thing is, and that's why these regions they allow the iodine solution remain in their original color. Why iodine solution cannot change to become dark blue already? Because you think inside, it consists of what kind of thing inside? What do you think? Because here, right, why iodine they can change color in this area is because this area, right, this dark blue color area consists of the starch. Okay, there's starch inside. 淀粉, okay, the starch. But later, when the times go by, the starch will be break down by the amylase. So the starch break down by amylase become what? Maltose. So that's why, will the iodine change in color if they detected the maltose? Will they change in color or they will still remain the color? What's the answer? If inside, yes?
No change
Hello?
in color.
Okay. If inside, you can see, if inside here, if inside here, okay, inside here, why the iodine solutions, right, they didn't change color? Remember the original color for the iodine solution is yellow, brown, or dark blue. Original color for the iodine solution is yellow, brown, or dark blue. Your answer is?
Yellow brown.
Yes, yellow, brown, correct. Original yellow, brown. So when the iodines get into the food, consists of starch, only they will just change in color, become dark blue, like this. Okay, like this. So that's why whenever you drop it, they just change in color until they reach this point already. Sorry, until they reach this point onwards, right? So even though you just drop the iodine solution, they will not change in color. Why? It's because these regions, they do not contain any starch. There's no starch in this region, okay? No starch in these regions. Because there's no starch in this region, right? So that's why when the iodine, they just in contact with the regions, with the areas, the iodine not change in color because there's no starch already. No starch. Why there's no starch? It's because the starch, right, is being broken down by amylase. What you say? Starch broken down by the amylase become what? Become the maltose. Yeah, become maltose. Okay, so that's why this area, which is this this is circle, this red color circle area, they consist of maltose only, no starch at all. So that's why when the iodines they contact in the area, right, they will remain in their yellow brown color. They will not change color because there's no starch. All starch only become maltose. So that's why when the iodines get in contact with maltose, so they will just remain in their original color, which is yellow brown. You get me?
Yeah.
So you can see iodine will only turn blue-black if there's starch present inside each of the rock tiles. So whenever it's not longer become blue-black, they refer to no starch is being present, but glucose is being produced. Okay, this glucose, right, is actually they refer to maltose. Okay, maltose. Because starch to maltose to glucose, okay, like this way. Maltose and glucose, they are both referring to the reducing sugar. Yeah, reducing sugars. Okay, so let's continue. Where is it? Just what I talk about. Okay, I asked about here. Okay, this one. Okay, plan an investigation to determine the effect of temperature on the breakdown of starch by the enzyme. So decide your method first. Okay, your method is you are actually doing what? Okay, you can use the Bennett solutions. Okay, because Bennett solution is a kind of solution they can detect for the concentration of the reducing sugar, right? They can detect the reducing sugar. So that's why you use the Bennett solutions added to the test tube to measure the reducing sugar produced at every 1 minute. So that's why you write the ingredients that you use and then how you can measure it. How you can measure the outcome. Ingredient, how you're able to measure the outcome. So, and then the concept they're being provided, because another thing is, okay, if you use any Bennett solution, right, to conduct the Bennett test, this Bennett, right, they should be conducted in the control temperature, which is around 70 degrees. Yeah, 70 degrees around that. It cannot be 100 degrees or it cannot be too cold. It should be 70 degrees around that. So that's why you need to do the experiment in the water bath. You need to insert the Bennett solution into the water bath. So you can warm it up, become 70 degrees Celsius. And then you can maintain a constant temperature. So this is the advantage of water bath, okay? It's for allow you to maintain a constant temperature. And then another thing is individual variables. Design individual variable, okay? You can say that the temperature, because you are actually study the effect of temperature change on the breakdown of starch, right? So how you're able to study your individual variables is you using the different temperature. So how you can use a different temperature, which is they referring to this one. You place a test tube containing the starch and the blend solutions in about 30, 50, 70. List down the figure. Okay, you can write as much as you could. Okay, you can write 30, 40, 50, 60 also can, up to you. About 3 or 4 figure for the individual variables. And then the constant variable is you can use same type of enzyme, same concentration of starch. This is very important because if you change in constant variables, it will affect the result accuracy. Okay, and then how is a dependent variable? So you can say this is dependent variables. The concentration of reduced sugars being produced. If you refer to the question just now, they ask for this one, breakdown of starch by the enzyme. Okay, after you break down starch by enzyme, what will happen? Starch will become maltose, then to glucose, right? So that's why the maltose and glucose is referring to what? It is referring to the reducing sugars. Okay, it's referring to this reducing sugar. So that's why how you're able to study your responding variables is you measure amount of product being produced. So you are actually how to measure the concentration of the reducing sugar being produced. So that's your answer. Okay, see, measure amount of your product being produced, and then how you're able to improve the experiments. Okay, repeat the experiments twice and wear the goggles. Wear the goggles when heating the solutions. So usually this last 2 sentences, right, is already fixed. Okay, just you can just copy the same sentence in your answer also. So that's why this 2 marks is already fixed. Okay, you need to score these 2 marks. So methods, individual variables, constant variables, dependent variables, and ways to improve the experiment. Just like this. Okay, 6 marks. Okay, this is when it test result. So if you don't have reduced sugars, you will have the blue precipitate. And then if you slightly increase, you are actually increasing the concentration for the reduced sugar, right? So you also increase the color. At the end, it becomes the bigger precipitate. So this is very important. And also, remember about this one, food test, the outcome for the food test, and also what is the color for the positive and the negative result. So for the starch, right, which is just now, okay, in order to test the starch in the food sample, you need to use the iodine test. So the iodine usual color is, the original color is yellowish brown. So if the positive result show there's a presence of starch, it will change from yellowish brown, become blue black. Okay, for the reduced sugar also, just remember, if you conduct any experiment related to the Venn-dick solutions, you need heating. How you heat them? Just put them in the water bath. Okay, water bath, and then you need to warm up the water bath with the Bunsen burner. Just require slight heating. Heating, not to boil them, okay? Just to allow them reach about 60 to 70 degrees Celsius. So that's why the original color for the Venn-dick solution is about blue in color. Okay, so that's why if you test, there's a presence of reduced sugar inside, and then it depends on the concentration of reduced sugar. Okay, so that's why the increase in color from green, yellow, yeah. Okay, and then this is safety precaution. Heating in the water bath, or you can say that wear the eye goggles or glove. Wear the glove also. And then protein fat test. Okay, also same thing. So protein is the one who change the color from blue to purple. Fat is from the clear solutions become white suspensions. Because if there's an increasing in the fats contents in the food sample, right, the white suspension become occupied in, more space. Okay, become more milky and more cloudy. This is called what? Cloudy milk emulsion. Yeah, emulsion test. Okay, emulsion test. And then for the vitamin C, right, they are actually using this DCPIP. So this DCPIP indicator is a kind of indicator. So that's why if there's a presence of vitamin C in the food sample, it will change from the original color, which is blue color, become colorless. So that's why just remember that out of those food tests, right, Benedict test is only the one required heating by the Bunsen burner in the water bath. Other food tests don't require any heating. Okay, so if you're able to, okay, for example, if you're adding the amylase to the starch, right, it will change the positive iodines become negative because the starch is being digested by the amylase, become maltose. Just remember the iodine only can change color in the presence of starch. That's all. And then for the Benedict test, right, it will become positive because now the glucose are being produced. Maltose glucose being produced. So they are belong to the reagent sugars. So that's why they change the color, okay, from blue color become orange color or the blue precipitate. Okay, that's all finished about this one. Okay, and then one document I haven't have time to go through with you, which is this one. This one. Okay, you can have a read. Okay, you have a read. Okay, this one, right? This is for you to have some, give you some idea. Because to answer this paper 6, you need to be, you need to be very detailed. How you're able to measure your the responding variable. Use what kind of apparatus. So this example of it. Okay, usually, right, the most common to be used is this one, measuring cylinder. String. Okay, string. And yeah. And then other thing is where is it? Okay. So this is just for you to have a look on it, okay? And then okay, so this one later you have to read, okay? Have a read. Yeah, and then this one, the independent variables. What's the common type of independent variables? So which is this one? Temperature, humidity, light intensity, airflow, carbon dioxide concentrations. So that's why how you're able to change the variables. Okay, because you already decide those parameters become independent variables. So how you can manipulate the variables? So this is a way. For the temperature, right? Because this point, it should be included in the procedure. You still remember, right? The 6 mark. 1 mark is given to procedure. So that's why you should write, okay, how you able to use any kind of methods to control your independent variables. For example, it's like this. Where is it? This one. The procedure, okay, they will ask you to write the methods, which is this one. Okay, where is it? Here. And here. See the method. Okay, the method. So you can include this one. In the methods. Okay, see, you are actually use the control water bath to maintain a constant temperature. Okay, so for temperature, right? See this one. Use thermostatically control water bath. Because you want to avoid the fluctuation of temperature also. If you don't have this water bath, right, maybe suddenly the temperature become what? Become 41. Maybe the drop become 38, right? So this will affect the result. So if you use a water bath, they can allow the temperature maintain a constant range. So that's why it's very important that you need to use a water bath if you decide the temperature become independent variables, because you need to manipulate the temperature. And then for the humidity, right, either you can cover the substance with or without a plastic bag. Okay, for example, I think they come up with this question before. They want to test about whether these plants, right, they will be affected by the humidity factors. So they will put out, okay, for example, right, okay, this is a plant, okay, this is a plant. This is one plant. Okay, this is another plant. Okay, another one plant is covered with a plastic bag. Okay, this is a bag. And then this bag is transparent. Transparent is what? Transparent means that they will not affect the photosynthesis because the light, they can still penetrate through the bag and reach the plant. But what happened to the humidity? Which one have higher humidity? is the plant will with the back enclose them, or it is the plant which is don't have the back enclose them? Which plant they have a higher humidity? A or B? What's the answer? Humidity refer to what? It referring to the yes, A. The answer is A, okay? Okay, you answer yes, it's because what? It's because that these plants, right, they will have the humidity, sorry, it will have the plastic bag trap the plants, right? So that's why it will cause the water vapors, okay, cannot be released out. So you think the answer is A. Okay, yeah. So the light intensity, right, you can use the lamp as different distance and the airflow, okay, whether you have a fan or not. And then for the carbon dioxide concentration, right, you are using different concentrations of the sodium hydrogen carbonate solution. Okay, this one, sodium hydrogen carbonate solutions is referring to the a kind of solution. This solution, right, they can release the carbon dioxide. Okay, this solution they can help to release the carbon dioxide. Something like a source of carbon dioxide. Okay, so planning for the experiments. So you can write down, use different concentrations of any kind of solutions, or use different temperature, something like that. Write down dependent and constant variable. Okay, constant variable, right? You can write about 3. I think you write at least 3 points. Okay, 3 points. Okay, 3 points. Some of the examiner ask 2 points, or some they ask 3 points. I will just confirm with you again. But write 3 points. Okay, 3 points. But if you're not confident, right, you write 2 points. And then method, okay, methods. Yeah, so the method, right? For the first few sentence, you write about what? You write about you use what kind of measuring. I mean, you are using what kind of apparatus? What kind of apparatus to measure the responding variables? And then later you can repeat this one. Okay, repeat the experiment to get more reliable result, or repeat the investigation for more 2 times, and then your safety precaution. Okay. Yeah. Conclusion. Okay, whenever they ask you to write a conclusion, right, it's something like this. Okay. For example, like the higher the quicker. Okay, you can use this kind of sentence structure. Just make it simple. And then for this one, okay, error and improvement. Well, which is that question. Okay. Just like I just showed you about. They ask about this one, right? How you able to improve it? This one, okay? The error and then the improvements. This is also a fixed question to come out in paper 6. So here's the answer. Okay, if you use the reagents to test and adjust color by eyes, you are using the colorimeter. If the variable was not controlled in the experiments, control these variables. And then monitor more than 1 sample at the same time. You do each one separately. Okay, so this is the way how you improve your previous procedure. If you're counting the bubble, right, it's better for you to use a gas string. Gas string is a more accurate way to measure the volume of gas. You want to prevent overheating to the test sample, so it's better for you to use the water bath, which is thermostatically controlled. And then for the length one, this one. Instead of calculate the final length, I think it's better for you to measure the change in the mass and the length to compare the final and the initial length. Okay, so this is all the sentence you can write down in your answer. Use the same string, cause conservation is better for you to use different string. Okay, different string. And then this one, right, you need to read the procedure carefully. And this one, just read this procedure very carefully, because the clue is just come from here. The arrow, you need to identify which method, which step, right, could have a room of improvements if you want to get more accurate result. Yeah. If you deal with anything about chemicals, so this is the precautions, or anything about cutting knife, which is the experiment, they got include the cutting knife, right? So cut away from the finger, okay? Be careful. Okay, so this is the way how you draw it. And then, okay, some of the fixed answer, right? Okay, why you should repeat the experiments? And then why you need to have this control group? Okay, control group refer to what? Refer to there's a one experiment, right? It's not one experiment. Okay, there will be another group, which is they didn't do anything. They just want to put as control, okay, because they want to reduce anomalous result. This is the thing. You know what I mean by this one? Anomalous result. Anomalous result is something like, okay, for example, right? And this one, when you draw a line graph, like this. Okay, this is a line graph. Okay, so this is your result. Okay, your x-axis and also the y-axis. So should be like this. Okay, constant. But there's one result out of the skew. So whenever you draw the line graph, right? Okay, so this one is called what? Anomalous result, or something called outlier. You know outlier? Which is the result, right? Is totally look not logical. We call it anomalous result. So that's why that's very important that if the question asks you why you need to repeat the experiments, it's because you want to reduce the anomalous result. Okay, just write this. Or why they ask you why you want to use a control groups. Because this is the reason. And then how you can result. Yeah, okay. If you want to control temperature, right, you use a water bath. Or if you want to improve the time, okay, the calculation for the time, right, you can take a smaller interval of time. Okay, and then when we draw the line graph, okay, so this is the way. See, you plot it and then you just connect it together. But you don't need to connect the outlier. Okay, if there's outlier. Okay, that's all for today. Okay, I think at this one, I have discussed finish with you or not. Okay, and then there's one more document, right? I think you can go through by yourself. This one. Hey, where is it? Okay, I think I go through all the document with you already. So, and then okay, the homework for today is you can attempt on the paper 6 by yourself. Okay, you try. Okay, you try to do this. Hey, where is it? You try to do this. 2025 May June, which is last year May. Try to do this. Okay, do this by yourself. Yeah, prepare a table, and then write down the variables. Okay, say conclusion. Okay, so this one conclusion, right? Conclusion for the result, you can use the method. Okay, the higher the what counting, the lower the what counting. Okay, see this one. Suggest 2 improvements for the methods used for these investigations. 2 hazards in these investigations. And then see this one, plan and investigation, which is the sixth step. Okay, the 3 variables and also the procedure and also the repeat the experiment after counting. So just copy based on that. And then this one also. Okay, they ask you to plot a line graph. So later you should plot the line graph. Okay. Okay, that's all. So see, what is meant by an anomalous result? They ask you about this one. The result which is become the outlier. Okay. Okay, you try to do this by yourself and then drawing also. Yeah. So they will come out the drawing. 4 marks. Okay, that's all for this paper 6. How many time for this paper 6? It's about 1 hour, I think. Yeah, 1 hour only. Okay, 1 hour. You try to complete in 1 hour. Okay, that's all for today. Bye.